What's up guys, and welcome back to another episode. Today, we're going to talk about a new subfield within biotechnology that has the ability to revolutionize the vaccine development industry. So, if you're ready, I'm ready. Let's break down single-cell genomics. Alright, as you've probably guessed by now, single-cell genomics refers to zooming into one particular cell and studying its specific genetic makeup. But wait, why would this be any better than just using bulk sequencing? Well, think of it this way. Assume each particular cell is a color. When you mix all the colors up and bulk analyze them, it gives you what the average color would look like. However, you lose the information which specifies each particular color and its special properties. Single-cell sequencing would be when you take a deep dive into each individual color, which exposes you to the specialized traits and features of that specific color. Now, let's take a look at the process behind single-cell sequencing. So, there's mainly 4 steps to this method: isolation, amplification, sequencing, and analysis. We'll take a look at each step in detail. First, we have isolation. As the name suggests, in this step, we isolate a specific cell from a tissue. This is done through a variety of different techniques, some being fluorescence activated cell sorting, or FACS, which utilizes lasers and fluorescent dyes to isolate cell populations based on their physical and chemical properties. Another technique used to isolate cells is laser capture microdissection, which utilizes lasers to dissect specific cells from tissues. For our second step, we have amplification, where we amplify the content of the genetic material in the particular cell. Again, there are a variety of different techniques for this, but the most well-known and widely used one is PCR, or polymerase chain reaction. So, PCR utilizes DNA segments known as primers, which essentially make multiple copies of a selected piece of genetic material by putting various nucleotides together. The third step of the process is sequencing, which is essentially where the different nucleotides of the genetic segment are read. This is usually done by complex machines known as next-generation sequencing, or NGS platforms. A few NGS platforms would be the Ion Torrent Proton and the Illumina Nova SEQ. Lastly, we have the analysis, where we use a software which would analyze and recognize any patterns or differences in individual cells. A few examples of software that provide these services would be the DESEQ2 and the Cell Ranger. Finally, let's discuss some of the pros and cons for single-cell genomics. Pros. It's helpful for understanding disease progression in specific cells. It helps build knowledge on physiological functions and diversity of cells. And lastly, it also paves the way for the advancement of targeted therapy. Cons of single-cell genomics. Firstly, it requires expensive equipment and a specialized team. Secondly, it comprises of the analysis of large amounts of datasets. And lastly, ethical concerns. Stepping into this field would possibly raise the issue of privacy and data ownership of this sensitive information. Alright guys, that's it for today. What do you think of single-cell genomics? Do the pros outweigh the cons? In my view, definitely. As long as we can overcome the data ownership ethical conflict, it is an easy win for single-cell genomics. If you guys enjoyed this episode, consider smashing that follow button and rate the show for more premium content. This is Biotech Breakdown signing out.